Scientists are now able to study the causes of human diseases and test drugs without any risk to human subjects using a human disease simulator. This tool allows for the examination of what propels the development of diseases. Envision a device that is smaller than a shoebox meant for toddlers, with the capability of emulating any human ailment across multiple organs. The device can also be used to test new medications without ever entering the body, thus causing no harm. The latest technology, known as Lattice, has been created by scientists from Northwestern University, which enables the study of interactions between up to 8 distinct organ cultures or human cell clusters for prolonged periods of time, mirroring the actual response of organs. The innovation is a significant improvement over the current in vitro systems, which are restricted to studying only 2 cell cultures at a time. The aim is to replicate the internal processes of the body to examine various phenomena. This includes investigating the impact of obesity on a specific illness, the differences in drug metabolism between genders, and determining the initial cause of a disease that progresses to affect multiple organs. Julie Kim, the head researcher and an obstetrics and gynecology professor at Northwestern University Feinberg School of Medicine, explained that the exact communication network within the body is still somewhat of a mystery. Typically, researchers study 1 or 2 cell types in isolation, followed by meticulous analysis. However, Lattice represents a significant step forward in this domain. The Lattice platform is a more realistic representation of the body, as it can replicate multiple organs concurrently, providing a significant improvement over previous research. The journal Lab on a Chip is set to publish an upcoming study on October 3rd that outlines the latest technological advancements in the field. The microfluidic tool comprises an assortment of channels and pumps that facilitate the movement of media, which is akin to simulated blood, between 8 wells. A computer that is linked to Lattice accurately regulates the amount of media that runs through each well, as well as the location and timing of the flow. Scientists can customize the contents of each well to examine a wide range of diseases or drugs by filling them with various types of organ tissue, hormones, diseases, or medications. Kim's laboratory is currently utilizing Lattice to investigate polycystic ovarian syndrome, PCOS, a medical condition that is identified by a hormonal imbalance and metabolic complications. Despite numerous studies, the root causes and mechanisms of PCOS remain a mystery. While it is commonly associated with ovarian health, it is important to note that other vital organ systems in the body are also impacted by this condition. According to Kim, the Lattice enables us to control and manipulate the organ that is responsible for the disease. For instance, we could use a PCOS ovary in one trial to determine how it affects the liver or muscles. In another experiment, we could investigate whether the high insulin levels linked to the disease prompt different organ systems to act abnormally. We have the ability to manage the tissues and arrange them in a predetermined manner. The contemporary approach to testing novel medications commences within a laboratory setting, where the experiment is carried out in a petri dish, in vitro. Following this process, it undergoes examination within animal models before proceeding to the clinical trials conducted on humans. Kim stated that there is no intermediate stage between animal experimentation and human clinical trials, resulting in numerous drug failures when tested on humans. Lattice may serve as the intermediate stage between animal experimentation and clinical trials by providing an opportunity to verify the safety of drugs that have passed animal experimentation on human tissues. This additional testing can serve as a final checkpoint before administering drugs to individuals. According to Kim, primary cell cultures grown in standard in vitro dishes have a limited lifespan. However, the Lattice system was specifically engineered to address this issue by supplying the cultures with fresh media or simulated blood and efficiently removing waste through the pumping of media through each of the 8 wells. This process extends the lifespan of the tissues significantly. Testing has been conducted for up to 28 days thus far, but the scientists aim to further extend this time frame. According to Kim, obesity is a significant hazard for endometrial cancer. She added that studying the impact of risk factors in a laboratory dish is not feasible because it necessitates long-term cultures. However, the Lattice platform allows for the examination of the direct effect of fat on the endometrium over an extended period. By utilizing this approach, early changes that occur in endometrial cells can be observed and studied. EVATAR, a miniature representation of the female reproductive tract, was initially developed by Teresa Woodruff, a former Northwestern faculty member and current co-author of the study. The second-generation product, Lattice, was designed to expand the range of diseases that can be studied, regardless of gender. Lattice is more cost-effective and user-friendly than its predecessor, and Kim anticipates that it will have a broad impact in both the research and pharmaceutical industries. Its creation is crucial in enabling scientists to safely and effectively test new drugs on the female reproductive system. Kim expressed that the goal for their project, EVATAR, has always been to create a user-friendly experience that does not require the presence of engineers to assemble and address any issues. The intention was to make the process as effortless as using a smartphone. Simply remove it from the packaging, power it on, and put it to use. This approach would allow researchers to focus primarily on acquiring data, rather than spending excessive amounts of time trying to understand how to operate the device within their laboratory environment. The research paper titled "The Lattice Platform, a novel microfluidic device for modeling physiology and disease across tissues," was a collaborative effort between the scientists from the University of Illinois Chicago, Rutgers University, and Michigan State University. The study's primary author is Hannes Campo, who is a postdoctoral fellow in Kim's laboratory at Northwestern University. To support our channel's growth and ensure broader awareness, kindly hit the like and subscribe buttons. This will help us reach more individuals and disseminate valuable information. Thank you in advance.